Hello, this is Digestive System video number 6. In this video, we're going to cover the small intestine. So this slide is showing the organs of the digestive tract that are located in the abdomen. So it's the lower GI tract. And everything except for the stomach. Stomach would occupy this position. And that's going to lead into the small intestine. Small intestine is a really long tube. It varies in different people. So, you see estimates of 30 to 20 feet long. There are 3 segments to the small intestine. The first segment is the shortest, a C-shaped segment. That's the duodenum. That leads into the next segment, the jejunum, purple tinted in this figure, leads into the last segment, the ileum. And then the ileum will connect to the large intestine. The small intestine is defined as the segment that spans from the pyloric sphincter connecting the stomach to the duodenum, and the ileocecal valve, which is the sphincter, just another name for a sphincter, that's controlling entry of chyme into the first part of the large intestine. This slide also shows some of the accessory organs that are associated with the digestive tract in the abdominal cavity. The largest is the liver, the gallbladder is associated with the liver, and the pancreas. Okay. What are the functions of the small intestine? 3 main functions, and this is where the bulk of the chemical digestion takes place. A little bit happens in the mouth, a little bit happens in the stomach, but when the chyme gets to the small intestine, it takes off. That's the main site. So chemical digestion is one of the main functions, and the enzymes for chemical digestion are coming from 2 main sources. One is from the pancreas, secretion from the pancreas that's going to go into the lumen of the small intestine. And the other is from enzymes that are embedded in the plasma membrane of cells in the mucosal epithelium of the wall of the small intestine. These are called the brush border enzymes, and the reason for that name will become apparent in the future slide. Right, that's one key function, chemical digestion. Second function is absorption. So the small intestine is the longest segment of the alimentary canal, and that provides a lot of opportunity for nutrients that are small enough from digestion to be absorbed across the wall and into the body. And in the small intestine, almost all of the nutrient absorption takes place. Third function is secretion. So there are what are called intestinal glands or intestinal crypts that are constantly secreting intestinal juice. Intestinal juice is going to mix with the chyme, and that's a third main function. All right, let's talk about the segments of the small intestine in a little bit more detail. So 3 main segments, starting with the duodenum. Duodenum is the entry point into the small intestine from the pyloric sphincter. Duodenum is relatively short, about 10 inches long, and it curls around the head of the pancreas. The duodenum is located in retroperitoneal location, so embedded in the back body wall. The stomach, on the other hand, is intraperitoneal. So at the pyloric sphincter, the duodenum, proceeds into the retroperitoneal position. The duodenum receives a lot of secretions, and some of these are from the accessory glands. The liver and the gallbladder secrete bile. The pancreas secretes pancreatic juice into the duodenum. And other secretions are from the mucosal membrane of the duodenum. For example, there are glands called Brunner's glands and they secrete an alkaline mucus, and this alkaline mucus is going to help to neutralize the chyme that's arriving in the duodenum from the stomach. The stomach is built to resist that acidic content that the small intestine is not, so it has to be neutralized. Those are some of the key features of the duodenum. The next segment is the jejunum. So this is the middle segment, and this is variation in length, but somewhere in the range of 5 to 8 feet long. So being relatively long and already primed with lots of enzymes, this is the primary place for chemical digestion and nutrient absorption. Unlike the duodenum, the jejunum has an intraperitoneal location, and it's attached to a mesentery, which is anchoring it to the dorsal body wall. The last segment is the ileum, and this is the longest segment. Again, variation, but it ranges from 9 to 11 feet long. And it terminates at the ileocecal valve, which transitions into the large intestine. Like the jejunum, the ileum is intraperitoneal and attached to a mesentery. The ileum is also a place of absorption. By the time it reaches the ileum, the chemical digestion is slowed down a bit. One of the key distinguishing features of the ileum is embedded in the submucosal layer are what are called Peyer's patches. Peyer's patches are basically groups of immune system cells. Since the intestinal lining is a vulnerable point for pathogens to enter into the body, the Peyer's patches are positioned at a location to defend against those kind of infections. So those are the three main segments and some of the features of those. Now, how is the small intestine specialized for absorption? Absorption is one of the main functions, and how does that work? One of the main features of the mucosal layer, and somewhat the submucosal as well, is there are some modifications, I guess you could say, or features of the inner mucosal membrane that vastly increases the surface area. An increased surface area makes more opportunity to make contact between the wall, the mucosal epithelium, and the chyme that's running through the small intestine. More opportunity for contact makes it more likely that those materials will be absorbed. So the three main ways that the mucosal epithelium is modified to increase surface area are shown on this slide. There are circular folds. We're going down this list from large to small structures. Circular folds of the mucosal and submucosal layers. Those protrude into the lumen of the small intestine. There are villi, which are finger-like extensions of the mucosal membrane, also extending in. And then at the microscopic level, there are microvilli, which are projections from the surface of the mucosal epithelial cells. So let's take a look at each of those and how they increase the surface area. So here's a segment of the small intestine, and we have cut away from the wall. We're looking at the different layers here, mucosal innermost, supported by the submucosal, muscularis layers, and it's surrounded by a serous membrane layer. If you look at the mucosal layer, it's not a flat surface. So it has these ripples or folds, and those are the circular folds. So they are go around the inside of the tube. Any chyme coming down this tube is going to bump into these, and it's going to slow down. And also there are valleys between the folds, and materials can get lodged in those, and that increases the opportunity for contact and makes absorption much more likely. All right, so that's kind of the macroscopic modification. You can see that with the naked eye, these circular folds. All right, it's sort of like a bunched up rug. Now we're going a little bit more microscopic, and here's a section of the wall, submucosal layer, and you can see the circular folds. This is going to run around the interior of the small intestine. This is a circular fold projecting in, and then at this level you can also see the villi. These are the villi, the projections, and these are the tips of the villi, all in the interior of the mucosal layer. So those are the villi. And now if we go even further magnification, this is what a villus would look like. Here are a bunch of them, and here's a villus in cross section. Okay, so this villus pokes out into the lumen of the small intestine. Now, here is the epithelium. The epithelium in the small intestine is a simple columnar epithelium. And all of these purple cells are what are called absorptive cells. Those cells are specialized to latch on to nutrients and transport them across the wall, the epithelium, into the lamina propria. The epithelium pokes out and the lamina propria also pokes out, so the finger-like projection. And then within the lamina propria are blood capillaries that can absorb anything that gets taken in across the epithelium. There's also, so the blood capillaries are represented in red and blue, right? So this would be an arteriole leading to a capillary bed, and this would be a venule draining from the capillary bed. There's also a vessel called a lacteal. A lacteal is a type of lymphatic capillary. So you have both blood capillaries and lymphatic capillaries. Now, a little bit about the lacteal. The name lacteal means it's milk-like, and the lacteals are lymphatic capillaries that are whitish in appearance. And that's because they are absorbing mostly lipids that are crossing in from your meal, lipids that get absorbed. The main destination for them is the lacteal. They will be circulated away through this lymphatic capillary. All of the other materials that are absorbed, breakdown products of proteins, of carbohydrates, of nucleic acids, will be absorbed into the blood capillaries and enter into the bloodstream. Okay, so two pathways. Lipids get absorbed into the lymphatic capillary, the lacteal, and all other materials get absorbed into the blood capillaries. Okay, let's see. Also in the epithelium of the surrounding the villus are goblet cells, so they're producing a lot of mucus, and mucus is produced pretty much along the entire digestive tract. Okay, if you look at the surface of the absorptive cells, these purple cells, and they're shown here, the surface that's projecting toward the lumen has these what are called microvilli. So the villi are the larger structures, and the microvilli are the same kind of projections, but this is at the microscopic level, the level of the surface of the cell. Okay. A few other things to point out on this slide. So this surface with the microvilli is called the brush border. It looks like a brush, right? Microvilli projecting. And those would be on all of the absorptive cells. They have these microvilli projecting out into the lumen. So that would be the brush border of the villus. Okay. And another vocab, the intestinal crypt or intestinal gland. So that's the epithelium that dips down into the lamina propria. And this is similar, but not quite as large as the gastric glands in the wall of the stomach. The intestinal glands or intestinal crypts are embedded in the lamina propria. And these have secretory cells that are going to secrete what's called intestinal juice. Talk about that in a second. Okay. This is an actual histological slide showing the intestinal villi. Here is the simple columnar epithelium with mostly absorptive cells, and you can see a few goblet cells there as well. And here's the lamina propria, and that's going to have blood vessels, lymphatic capillaries embedded in it. And so those are, you can see these are crowding the inside. And they're sort of swaying in the current of the chyme flowing down the tube. All right, and here the opportunity for contact with materials, just the huge surface area that results. Now, if you look at just the segment of the villus under high magnification, so this is an electron microscope image, you can see the brush border. So those are the microvilli, and just a huge increase in surface area there as well. Okay, so huge modification of the intestinal epithelial layer of the mucosal membrane is to vastly increase the surface area of that structure. It said, let's see, going back here, this summary slide mentioned that the sum of the effect of all three of these modifications increases the surface area of the tube about 100 to 200-fold over that of just a flat surface tube. So that's a huge increase in surface area. And the estimates are, there's a lot of variation, but in general, estimates are this is about half the surface area of a singles tennis court. Okay, so huge opportunity for materials to be absorbed. All right, so absorption is one function. Also digestion, another function. Secretion is a third. So let's talk about that. The main secretion of the small intestine is through the intestinal crypts or intestinal glands here. So there are secretory cells that are producing this secretion. And what is the secretion? It's mostly water, some mucus. There are goblet cells there as well. And there are some digestive enzymes as well. Not very much, not a huge source of digestive enzymes, but there are some. Normally you produce about 2 liters of this per day, and this is stimulated by food in the stomach or arriving in the small intestine. The acidity that hits the small intestine stimulates those secretions. The main function of this very watery mucus type secretion is to dilute the chyme. That's going to make it easier to absorb and also easier to propel down the length of the tube. All right, final topic is how is motility carried out in the small intestine? And motility is through 3 main things. The main thing that's driving motility is peristaltic contractions. So the small intestine is a really long tube, right? And it's very twisty and turny. And so in order to prevent stuff from getting trapped or stuck, peristaltic waves are all constantly running down that tube, pushing forward the chyme through the tube. There is also segmentation, which is kind of alternate contraction of nearby segments. And this is sort of creates a back and forth movement that helps with mixing of the contents. Finally, there's what's called the gastroileal reflex. And this is what happens when food arrives in the stomach. So remember, there is the stomach has a way of storing food and allowing small bits of that to pass into the small intestine. So whenever there is movement of food into the duodenum, that triggers this gastroileal reflex. The gastroileal reflexes opens basically the ileocecal valve, allowing food to that's going ahead in the small intestine to enter into the first part of the large intestine. So I'm going to end this video here. Next video covers the large intestine.